Okay, the second video is actually the continuation of the fourth and last method, which is the study of nuclear RNA. So it involves lots of other kinds of RNA. So one of them is the snRNA or small nuclear RNA, which is involved in RNA processing reactions. And this study has served as a model system to, of course, give further study on transcriptional machineries. And this is, of course, done by using factors and activators for regulation of transcription. As the name says, okay, so this study is done through transportation across membranes from nucleus to cytoplasm, okay, and this is done during transcription or post-transcription. So basically, this is how it is done. So I'm not going to dwell too much on this diagram because there is one activity for you guys to do. But simply said, okay, it involves RAN-GTP, okay, a protein, okay, that helps to basically move around, okay, the cargo, the load, okay, to be transported, okay, from cytoplasm to nucleus or nucleus to cytoplasm, plus the RAN-GDP. So RAN protein is involved. So whatever to be transported, okay, is called a cargo. Okay, so regardless of any types, okay, of DNA or RNA or anything, okay, to be transported is called cargo. And also it involves a molecule of a factor called importer or exporter. So the mechanism is actually very specific. Anything that is to be transferred from the cytoplasm to the nucleus, okay, the process is called import. So you have an importer. From the nucleus to the cytoplasm, okay, we call it as export and not the other way around. So this is actually a very precise mechanism. Don't ask me why. Again, it has to be precise such as this. Okay, you can actually read on about it because how it comes to be is actually very kind of like it's quite obvious, okay, in a sense, okay, if you look at how the cell is being built up, again, nucleus and cytoplasm. So if you could understand the concept, you can actually see, okay, why, okay, from here to here is export, one here to here is actually called import, right? So now, okay, but simply said, okay, so we know that basically all the replication, okay, is done in the nucleus, okay, so for transcription, okay, and also later on, okay, for translation, it has to be done in the cytoplasm, so it has to be exported. So to be exported, okay, your cargo, okay, have to have, okay, the exporter, which is one of the factor, plus the RAN-GTP to be across or to be taken across, okay, the membrane. Now, without these two, okay, your cargo will not be allowed to pass through, kind of like at the border control, okay, whereby if you were to enter Malaysia or from Malaysia to enter into another country, okay, whereby your passport, okay, will be checked. So those kind of mechanism, I'm actually giving an example so that you can relate to it. So now, once it has gone through this, okay, and they have checked, okay, that, okay, you have all the necessary documents, which is exporter plus the RAN-GTP. So they allow you, okay, to cross, okay, to the cytoplasm from the nucleus. So what happens over here, okay, your cargo will be dissociated, okay, from the RAN-GTP and the exporter. So what happens is that, okay, the exporter, okay, will be transferred back to the nucleus because you need it, okay, to transfer anything from the nucleus to the cytoplasm. So it has to go back, okay, to the nucleus. So exporter has to have the accompaniment of RAN-GDP. So it cannot go back on its own. Alright, so okay, RAN-GDP, okay, will accompany exporter to go back to the nucleus, okay, for the next use, okay, of transporting, okay, another cargo from nucleus to the cytoplasm. So over here, RAN-GDP, okay, GDP, sorry, okay, will basically okay, will be converted to RAN-GTP, triphosphate, from di to tri. So anything that is imported, okay, from the cytoplasm only need importer. That's it. Why? It's actually a very complex explanation, so I'm not going to further dwell on that. But okay, it's just your cargo plus your importer, okay, whereby, okay, over here at the membrane they see that, okay, you have an importer, so the cargo is allowed to go back in. Again, similar process, the cargo will be dissociated, okay, from the importer. But okay, what happens over here is that because, okay, to import your cargo from cytoplasm to nucleus, you need an importer. Similar mechanism, importer needs to be transferred back, okay, to the cytoplasm. So this now has to be accompanied by the RAN-GTP. So RAN-GTP will accompany your importer, okay, to be transferred back to cytoplasm. It will be dissociated, and of course, it will return back. The RAN-GTP will return back to the nucleus. So these are the kind of mechanism that occurs, okay, across the membrane. Now, what happens is that there is a lot of regulatory RNAs involved for the nuclear transfer. So we have various kinds of RNA molecules, long or small, okay, depending on the sizes, and they play a role or play roles in inhibiting protein coding genes expression. So this is done by interaction with specific gene or mRNAs. And again, just to cover on this, okay, it's actually a very long process. So we're just going to go very simple or summary of it. So protein coding DNA basically are only about 1% of human genome. Did you know that? So about 3/4 of human genome basically are transcribed into RNA, not all of them. So regulatory RNAs would regulate gene expression both at transcription and also post-transcription. And this would involve three major classes of small RNA, which is the microRNA, okay, MI, small interfering RNA, SI, and also PV interacting RNA, PI. So now what happens is that we know that gene expression could be inhibited by regulatory of the RNAs via induction of mRNA degradation, blocking of mRNA translation, or inhibiting the transcription. And this is done by the work of the three RNA molecules just now. So we'll see it in a while. But just a rough kind of like comparison of the three major classes of these small RNAs. So you can just read on about it in your notes. Now, for the first one, which is the microRNA, so a bit about it is that it was first discovered, bound to mRNA producing gene in the nematode, small animal. So binding can occur either in full or partial complementarity, and single mRNA basically can bind to multiple mRNAs at different target sequences. And this will result in inhibition of expression of many different target genes. So what happens over here is that we know that microRNA is basically a small molecule, as you can see from over here, or probably not really that obvious, but it's actually very small, and it is a small non-coding RNA. So basically it functions in RNA silencing, very similar to gene knockdown, and also during post-transcriptional gene expression. So apart from that, it also functions as a guide via base pairing because of the complementarity mechanism, and this is done within mRNA, and this is done or said to be done in a negative regulation. So this process would determine which silencing mechanism will be involved, and usually microRNA is being used for silencing. So again, what happens is that it will determine how it is going to be cleaved and also repressed. So it will most of the time destabilize the mRNA by either cleaving or repression, and this is done when it binds to the 3' UTR. I think it's not shown here, but usually it has to bind to the 3' UTR, which is basically the one that is untranslated, and of course it will repress protein production because a translational will be repressed. Now, the second one is the small interfering RNA. So what happens is that for the siRNA, or sometimes referred to as short interfering RNA, of course it's small in size, so it is processed from double-stranded RNA as defense mechanism against viruses, and the first study was basically done in virus. So it is formed when dicer protein cuts double-stranded RNA into smaller molecules, so you get single-stranded RNA, or also called as, of course, your small interfering RNA. So it will then bind to the identical sequence of original double-stranded RNA to promote degradation. And of course, once it binds to it, as you can see over here, so it inhibits viral gene expression post-transcriptionally, of course, and it has been widely applied to silence genes. So what happens over here? I think I have, what happens over here, it also involves a dicer, cleavage by the dicer from the double-stranded. So this is the one that basically make sure that the small interfering RNA will be produced, and it binds to the complementary transgene or the mRNA to degrade for it, and of course inhibiting their expression. Again, similar more or less mechanism. Now, what you see over here, is the dicer producing the siRNA, and before any silencing could occur, it actually form an RISC. So RISC is actually RNA-inducing silencing complex. So of course, it is a multi-protein complex, which is also kind of referred to as a ribonucleoprotein, and it is basically used to incorporate one single-stranded mRNA or sometimes like a double-stranded siRNA, as you can see over here, and this would call the silencing of the mRNA or halting the transcription process. Alright, so the third kind of RNA is the pi RNA or PV interacting RNA. So how they come about with these names, okay, don't ask me. So it's actually very complex. So PV interacting basically is the largest, okay, small non-coding RNA. So as you can see, okay, these are some of the sizes. So originally identified in Drosophila germ cells. So it is processed similarly to microRNAs and siRNAs, but shortened at both 5' and 3' without involving dicer protein. So no dicer protein is actually involved over here. So how, well, you can read on about it, but basically pi RNAs inhibit the activity of DNA transposons that move around the genome by binding to the transposon RNA transcript to induce degradation. So it also degrades, but what happens is that it functions as RNA silencer. So this is done via formation of RNA-induced silencing complex, the same RISC as well. Okay, and it also interacts with PV protein. So PV protein is RNA binding proteins, and it is also kind of like function as a guide for PV protein to cleave target RNA, and also cause it to be shortened, 3' shortening. So apart from that, it also promotes heterochromatin assembly and also sometimes methylate DNA. Okay, so as you can see, I actually should show this again when I was explaining just now. Alright, so now I would just give it straight away here in the video, okay, but probably I would also put in your e-learning. So I actually need you to do an activity on nuclear RNA export pathway for you to understand better. So of course it would involve group discussion. So you could use the same group for the two-component system, is it? Okay. So I want you to discuss on nuclear RNA export pathway of snRNA, mRNA, tRNA, and rRNA. So SN is small nuclear, okay? And basically mRNA, you know, okay, which is mRNA, tRNA, the ones that involve for the transfer, which is for the translation, and rRNA, ribosome. Now each group, okay, not of 6, okay, because last time we have a large group, okay, so each group of 4, okay, around 5 people, because I think we have 2 extra people, should provide a summary of designated pathway and how it can be used to measure or visualize transcription rate or level if you could find some information, along with general illustration or diagram. So that means that you need to come up with your own diagram. No copy and paste from the internet directly. So each group will then present their findings to the peers, but this basically I will see whether it could be done or not, but probably not this week, some other weeks or towards the end of the week, whereby when we do similar thing as we did during the procreator, towards the end we basically discuss everything from the beginning up until the end. So again, you are to make a video, basically make a good diagram on how each and every one of these is being exported, and then, well, we are going to basically see what you found out. So during the class or in your e-learning, I will basically assign which group will do which of this RNA. So obviously there's going to be 5 groups, I think. So probably I will add another one. Because since we only have 4, so I will add another one. So don't worry, look up in your e-learning for further instruction.